Good afternoon. Shall we get ready for our fourth and last session? So I'm a cancer epidemiologist, so therefore I'm very pleased to see the last session today, which is 15 years of the bladder cancer prognosis program, which Rick has been leading on for many years. And so he's going to start off with telling us a little bit about how they got to the bladder cancer prognosis program and some of their outputs. And then we have several people of the team from Birmingham who have moved on, some of them since as well, to tell us a little bit more. And I'll announce their names when we get to it. So Rick, please tell us more about this program.
Thank you. So the bladder cancer prognosis program, really this is hopefully just a quick talk, because I think the following speakers are going to be far more interesting than I am, but really just to highlight what I call the life cycle of a cohort study. So based around the bladder cancer prognosis program. So this was originally led by my colleague at the University of Birmingham, Professor K.K. Cheng. I was part of the team that started writing the program application, and we started writing in 2004. And actually, we started writing round about the time that I was writing up my PhD, and I came under a certain amount of pressure to start writing this program grant instead of writing up my PhD, and I took a bit of a gamble and went with this program, and essentially it has provided a generation of cancer research both for me and my team and lots of other academics around the world. So although I'd finished the laboratory work for my PhD in 2003, I wasn't actually awarded my PhD until 2007. So, and actually interesting to see that some of the output being cited by Ben in the previous talk. So we were funded by CR UK, over £2 million, with 5 original objectives: to assess the effect of lifestyle factors such as smoking, dietary habits, fluid intake, environmental exposures on the recurrence and progression of non-muscle invasive bladder cancer, to study health-related quality of life and its relationship with recurrence and progression, to establish a collection of bladder cancer biospecimens comprising blood, tissue, and urine, and to study the predictive effect of molecular markers with regard to recurrence and progression. And finally, we nested within the cohort was a randomized controlled trial of oral selenium and vitamin E for preventing recurrence and progression, a study called Selenib. Where are we at the moment? Well, between 2005 and 2010, which was our original funding from CR UK, we recruited over 1,500 newly diagnosed bladder cancer patients from around the West Midlands. They have currently a median follow-up of over 5 years, and we're just in the coming months about to start a sort of 10-year sweep of all the survival outcomes. We have sets of frozen tissue, paraffin-embedded tissue, blood and urine from around 1,000 patients. And that's really fueled much of the work, the laboratory work that we've done with regard to urinary biomarkers, transcriptomics and molecular classification, virtually a generation of laboratory-based translational research. Over 60 publications have stemmed from the program, and all our original objectives have been achieved. I suppose I say here the second phase translational objectives underway. You know, they've been kind of underway over the last 10 years. We've completed a number of collaborative genome-wide association studies. More such studies are ongoing using the same research data and outcomes, and you'll hear more about that shortly. As I said earlier, we've devised, developed, validated, and commercialized a diagnostic urine test for bladder cancer. And we're working hard on better molecularly stratifying non-muscle invasive bladder cancer. We've made quite significant strides as part of international groups working with others. There's a lot more to come, which again, you're going to hear about later on. Really, I just wanted to present, this isn't, this just shows you the publications over time. And obviously there's been quite a slow burn, if you like, you know, that we'd only, by the time we'd finished recruiting patients and our funding ended, our CR UK funding ended, we'd published very little. And actually over time, that has increased significantly for, you know, for at least 10 years after we finished recruiting patients. And really that is as the biospecimens that we have collected have gained in value because we have mature outcome data to accompany those biospecimens. And a lot of the research platforms that we're using have come down in cost very significantly. So we can now undertake very sophisticated multi-omic, genomic, proteomic analyses on these samples cost effectively, which we couldn't have done 10 years ago. And also the program has been a great vehicle for training academics and clinical academics. And so 7 PhD students to date have been through the program, and some of them have even survived and are here today. And we've got 7 more, 2 more due to join us later this year. And really, you can kind of map the number of PhD students we've had onto the publications. I'm trying to think, is there a pointer on this? Is there a pointer? There is somewhere. Oh, yes. There we go. So, yeah, PhD students. So, here we will have Fritz, Sylvia, and Duncan, and you'll hear from Fritz and Sylvia later. And then later joined by Nadia, who you'll hear from later. And then Nada, Birmingham Nada, as opposed to Aberdeen Nada, joined. And then Sylvia and Fritz and Nadia had already left. Then we're down to just Nada. And then we've got Ming Hao and Faisal adding there. So I'm hoping that it looks as though there's a bit of a sort of lull in publications here, but as the PhD student numbers have come back to a good level, I'm hoping to see more peaks in the future. So really that's kind of the message, really, that cohort studies take a long time to warm up. And important outputs and impact may only be apparent more than 10 years after their initiation. They also represent great opportunities for training the next generation of scientists and clinician scientists. And in essence, funders should be aware of this phenomenon and be patient about the outputs that they expect. And that's all I'd like to say before handing over to the team who've done some great work. Thank you.
Thank you, Rick. So I'll leave the questions then for the next speakers. And so next we'll have Frits van Os from the University of Maastricht, who will tell us all about the work they've done in the bladder cancer prognosis program on smoking and bladder cancer. Thank you, Frits.
All right, thank you for the introduction. So yeah, just to add on that introduction a little bit and to explain you why this tall Dutch guy is standing here and happy to be back in the UK after, I think 3 years ago is the last time I was here. Now I'm working as a clinical epidemiologist in a teaching hospital in the Netherlands, but indeed, as Rick has already introduced, I will walk you through my PhD project and how I survived the program. I did a joint PhD program, so I was both at University of Birmingham and Maastricht University. So I will try to walk you through my whole PhD project in 15 minutes, but don't worry, I've done it before. In the UK, you defend your PhD thesis in a closed room during a viva. In the Netherlands, you also have to defend your thesis, but then in a room full of people, and you have to start off with a 15-minute talk about your PhD. So I've just translated it to English, and I'll try to give that overview again. So that's also where the layout comes from. You make sort of a fancy booklet of your PhD thesis in the Netherlands. That's a bit different from here. Just found it fun to show this. So I won't go over all the chapters to keep it to keep the speed in the talk a little bit. So I will focus on these ones. I've done research on 2 parts of smoking and bladder cancer. So the first is smoking and bladder cancer risk, of which we already know quite a lot, also when I started in 2014. And then the second part is smoking and bladder cancer prognosis, for which, at least at the start of my PhD project, less was known, especially about smoking cessation after diagnosis, but we'll come back to that in a minute. So you saw in those chapters that I did a few meta-analysis, just as a quick reminder, or for those of you that aren't familiar with this, what is it? What you're doing, you're not generating new evidence, so you're not doing a new study or recruiting new patients, but you're actually summarizing and weighing the evidence from studies that have already been performed. So you use more studies, larger sample size, different populations, so it gives more certainty about the association that you're looking at. But again, no new evidence is generated, so you can only do it for topics that have already been studied a lot. Well, luckily for me, smoking and bladder cancer has been studied a lot already. So I started out my PhD project to see actually what was known already, to update this meta-analysis. So we found 89 observational studies that investigated smoking and bladder cancer risk. In total, that were 57,145 bladder cancer patients. So that's quite a few. They were from all over the world. So that means you also get a very large table in your article, because you have to summarize all those papers a bit, so that wouldn't even fit on one page. One of the important things I think that we took from this meta-analysis was a dose-response analysis that we did. So we pulled all these studies together. Let's start in the bottom right corner. I think smoking cessation is the most interesting one. You can see here, well, this little bubble, that's likely an artifact of statistical analysis. I like to go on for that for hours as an epidemiologist, but I won't bore you with all the statistical stuff. You can just assume this sort of smooths over. But what you can see here, even people that have stopped smoking 30 years prior to diagnosis, they are still not at the equal risk of 1. So you're sort of dividing the 2 risks by each other of people that have never smoked compared to people that have stopped smoking in this case. They never actually get back to that same risk of people who have never touched a cigarette, or maybe just one at Christmas. That's what someone wrote at one of the questionnaires. So they were very, very honest. So that's interesting. Another interesting thing here, also for the people who are interested in the molecular processes behind smoking and bladder cancer, is that you see sort of a plateau in the graph there, right? So if you smoke 20 cigarettes and if you smoke 30 cigarettes a day, the risk compared to someone who has never smoked that's always the reference group, is more or less the same. So it seems like there's that sort of an effect that if you already smoke a lot, the damage is already done. What we confirmed and what we already knew from previous literature, that smokers have about a 3 times higher risk of developing bladder cancer compared to people who've never smoked. And that smoking behavior, if you look at the specifics, that especially stopping smoking significantly influences the risk of bladder cancer. But I want to look in this a little bit further. I wanted to look at the interplay between duration and the number of cigarettes that people smoke, right? Because does someone who has smoked for 5 years and 2 packs a day is at the same risk as someone who has smoked for 30 years but just 2 cigarettes a day? That's what I want to investigate too. Well, for that, we had another study from which we had the actual data from 15 case control studies around the world. And we did an analysis on the risk patterns in these different smokers. So we looked at pack years. Pack years is a combined measure. If you smoke 1 pack year, that means that you smoke 1 pack per day for a year. So if you smoke half a pack, you have to smoke that for 2 years to get 1 pack year. So what we did here, it's a really nice little model. It took me a year, I think, to come up with this and some help with the statistician from Utrecht University. But what we thought of is a analysis where we looked within the pack year category. So let's say within a group of patients that smoked 10 pack years. So that could either be people that smoked 10 years a pack a day, or people that smoked less cigarettes for a longer time. So what we see here in this model is the excess risk, so the extra added risk that you see in the model if you look at the number of cigarettes that people smoked per day. So the line goes down, so that means that people within the same pack year category that smoked a lot of cigarettes had a lower risk compared to the people that smoked longer but less cigarettes per day. Just to put that into normal people words and not epidemiologist words, that means within equal pack years categories, smokers with a longer exposure, so the longer you smoke, the higher the bladder cancer risk becomes. And so if you compare it to a heavy smoker with a shorter exposure, they're at a significantly lower risk than someone who smoked less cigarettes over a longer time. So, well, stopping smoking at any point makes a lot of impact, and the most impact, actually. So that's, in short, what we learned from the studies that we did on smoking and bladder cancer risk. So looking at prognosis. Well, prognosis can mean different things, right? So within the bladder cancer prognosis program, we have different endpoints. So now Rick told us we have the 10-year outcomes and also the mortality. But during my PhD, we mostly looked at the recurrences of bladder cancer and not so much yet at progression. So from non-muscle invasive to muscle invasive and dying. So I'll focus on the recurrences and endpoint. Again, I started with a meta-analysis. So looking at the studies that were already published. And we could see here that people who were former smokers at time of diagnosis, they were at a slightly higher risk of a recurrence within 2 years compared to someone who had never smoked. It was not statistically significant, but if we looked at people that were still current smokers at time of diagnosis, they were at a 1.27 times increased risk of recurrence. So as we call that in epidemiology, not that large of an effect size. It's not like a 3 times higher risk, 1.27, but still we were able to show this in this meta-analysis. So that's how we got to the study that we did in the BCPP study. This was already introduced by Rick, right? It's the same study that we're talking about. What we did there is asked the patients about their smoking behavior at diagnosis, but also at follow-up, which is unique, I think, because there were not that many studies. There's a few now, but not that many studies that looked at stopping smoking. So I was really curious to find out what happened there. So we know the duration of smoking is important, but what happens if you advise people to stop smoking? Well, unfortunately, I would say, of the 174 people that smoked at diagnosis, only 24 stopped. This was regular clinical practice, right? We had no intervention in the urology clinic specifically to advise people to stop smoking. So I've looked it up in literature in preparation for this talk. If you do interventions, you can get this up to 50 to 70% in different populations. If you do an intervention with your urologist or your GP to stop smoking. But in regular care, this is a normal figure. A large CPRD study published in 2017, their smoking cessation rate in the bladder cancer group was 21%. So there's some work to do there. But yeah, also the results from the meta-analysis, so this slightly increased risk, we couldn't replicate that within our cohort, likely because the association is less strong. So there's likely some association, but not a really, really strong one, and then you need a huge study to show that. Nevertheless, again, stopping smoking before diagnosis seemed to be associated with the decreased risk of also bladder cancer recurrence. So if you stop earlier, it decreases your risk of getting bladder cancer at all, but also of getting it back again if you are still, well, unlucky and get bladder cancer anyway. Well, it's safe to say that smoking is one of the most important risk factors for bladder cancer, and then especially for those who have smoked over multiple decades. Well, the evidence is still a bit inconclusive about stopping smoking, but there are more prospective studies. Luckily, this call that Rick made about funders giving money to these prospective studies has been heard a little bit already. So we're getting more data on it. So I'm looking forward to seeing what happens in the future. But my advice for now would be try to help as many people as possible to quit smoking earlier before they get bladder cancer already, to reduce the burden on patients' lives, but also on the healthcare system as a whole. Thank you.
Thank you, Fritz. Any questions for Fritz? Well, I'll ask you one whilst people think about their questions. So we know that with smoking and bladder cancer, it's 4-aminobiphenyl, I think, is one of the components in the cigarettes, which is very much, if I remember correctly, it's in the black tobacco.
Yeah.
So have you done any work around like blonde versus black tobacco and over time, because these things have changed?
Yeah, we, thank you. We also did a study that was, I think it was chapter 3, and that I skipped for this presentation. And we looked at different types of tobacco. In our study, we couldn't find an association, but there were other studies from Italian groups that did show indeed that the black tobacco constitutes a higher risk of getting bladder cancer than the blonde tobacco. Yeah. Yeah.
Any other questions? Yes, there is one in the back.
Thanks for the great presentation.
Thank you.
Did you see any associations between sort of starting time of smoking and onset of disease or diagnosis of disease?
Yeah, we looked at that too, indeed. So you mean whether you start at your 20s or at your 40s or?
Yeah,
something
let's
like
say
that.
somebody starts smoking when they're 20 years old.
Yeah.
You know, will they get bladder cancer earlier
Mhm.
than, say, what you'd expect in a general sort of older population?
Yeah, we didn't look specifically at the age of getting bladder cancer, because that's a little bit of a different question. But we did look at the age starting smoking, and then that was sort of diluted by the effect of the total duration of smoking. So you saw that most people that started younger were also at a longer duration, so we're in the higher risk group. But I didn't do a separate analysis on whether they were also younger when they got bladder cancer, to be honest.
Thank you.
We'll have you back to do that.
Yeah, yeah, that's a good idea.
Just out of pure curiosity, what about bystander effect, you know, for people who don't smoke themselves but live with smokers?
Yeah, that's the other chapter that I skipped. Passive smoking, also in children, for example, of where their parents smoke. There we found similar associations as with the recurrence. So it was not 3 times higher risk, but 1.5. Some studies 2. Yeah, so that's also an important factor to consider. Yeah, so.
There is more.
Oh, yeah.
You obviously looked at a lot of recurrence. Did you look at markers of lethality of relapse? And people who have bladder cancer, is smoking associated with greater risk of lethal recurrence or progression?
Yes, thank you. Well, I think now, maybe I should just return, Rick. I think now we could look at this properly within BCPP with the 10 years outcomes, but I've looked it up in the literature too, and in the meta-analysis. There's no definite studies that really isolate this effect. It's mostly if you also quit smoking that it helps in the perioperative and also when you have chemotherapy with preventing complications. Yeah, it's really with mortality as an outcome or bladder cancer related mortality, there's not that much evidence yet. So overall mortality, yes. Bladder cancer related mortality, it's still a bit unclear.
And I may as well complete it. What about men versus women?
Also interesting question. So what we see in general is if you look at the population attributable risk of smoking in predicting bladder cancer, that proportion is even higher if you look just in men than in females. Well, also in that model, for example, with that duration extra added risk, that association was even stronger in males than in females. So that's quite interesting to dig into a bit more too. And what's that actually, well, happening in the body? Yeah.
And whilst we wait for the mic to go to Nada, I'll have one more question too. So, because you talked about the overall mortality, so obviously in epidemiology, you struggle with the concept of competing risks, because smokers obviously have a higher risk of dying of cardiovascular disease, diabetes. So therefore, they may die of this before they would get diagnosed with their bladder cancer. So have you done anything? Maybe your follow-up wasn't long enough.
Yeah, exactly. So. Then I'll have to just refer back to the call that Rick did for good prospective studies in which we can account for the competing risks. Indeed, that's something we couldn't do yet, but yeah.
When you were assessing the smoking status of all these patients, was it just cigarette smoking, or did you consider other forms like pipe or tobacco chewing or even e-cigarettes? Yeah, the
results that I showed here were cigarette smoking, but we also did analysis separately for pipe and chewing and et cetera, et cetera. The ones for cigarette smoking were the largest sample sizes, the most pronounced, but it's sort of the similar associations for the pipe and tobacco, chewing. Yeah.
Yeah, that would be my follow-on question. Did you see any difference?
Yeah, well, I think that also relates to the meta-analysis on risk. What we saw there were also really big regional differences. So studies from Asian countries, there the risk was only 2 times as high compared to the never smokers, while in the European American studies it was 3 times as high. So I tried also in that type of cigarette analysis to account for this, because the cigarettes are heavier in Asian countries, but we couldn't get the data. It was restricted. Yeah.
Thank you. Is there another question, Anna?
That's just a comment from Nick James on the chat. Yeah. A bit of a tongue-in-cheek comment once from Jim Cato that smokers did better post-op as they had to mobilize early to go outside for a fag.
Yeah. Yeah, physical activity, that's another interesting one to consider. Yeah.
Thank you. Thank you very much, Fritz. So then we'll move. So then we'll continue with your colleague Sylvia Jochems, who is now at the University of Lund, and she will tell us more about diet and bladder cancer. Thank you.
So hi, first of all, thanks Rick and Mieke for having me, for giving this, that I can give this talk. So yeah, currently, well, before actually I did my bachelor in biochemistry, and I actually worked on the lab in biomarker research. But then I thought like, oh, you know, it would be very interesting to combine lab with like more epidemiological research. So after that, I decided to do a master's in health sciences with like the focus on clinical epidemiology. And after that, I finished that, I got the opportunity to work as a PhD student in a joint PhD project, just like Frits. That is like a program between the University of Maastricht and the University of Birmingham. And my thesis was about diet and bladder cancer risk and recurrence. Well, at the moment, I work at the University of Lund, but that was because after my PhD, I was actually looking for a postdoc somewhere in the Netherlands or in the UK, but then this great opportunity came by in Sweden, so I just like packed my bags 2 days after I defended my thesis here, and I moved to Lund. So at the moment, I work as a postdoctoral researcher at the University of Lund. I switched slightly from bladder cancer to prostate cancer, so still in the urological cancers. But, and I do want to mention my PI of the group that I work now for, because she is one of the best epidemiologists that I know in cancer research. And I'm not going to bore you with our results from our project, but my project on lifestyle factors like smoking and body mass index and waste and prostate cancer. But I do want to mention this one because I'm very proud of it. It's the latest paper that we have written on smoking and prostate cancer risk and survival. And 5 days ago, it has been officially accepted by European Urology for publication. But now on to bladder cancer. But before I go to diet, I quickly want to mention Stanley Teleka. He was a PhD student also at Tanya Stock's group. And his project, his thesis was actually on metabolic factors and bladder cancer risk and mortality. The main focus of his research was on blood pressure, and also very nice publications of that came from it. So he looked at if increased blood pressure could increase the risk of bladder cancer. And in fact, he found that high systolic blood pressure does increase the risk of bladder cancer, overall bladder cancer. And this was then, we could find that in conventional analyses like these, but also in Mendelian randomization analysis. And further, when we would separate bladder cancer and non-muscle invasive bladder cancer and muscle invasive bladder cancer, we found actually that systolic and both diastolic blood pressure could also increase the risk of muscle invasive bladder cancer, and for systolic blood pressure also for survival. And also our latest article that we published earlier this month on bladder cancer, we actually found that individuals with a slow acetylation NOT2 variant would most benefit from smoking cessation to prevent lethal urothelial cancer. But now on to diet and bladder cancer. So during my thesis, I looked at bladder cancer risk and recurrence. And for bladder cancer risk, I used data from the BLEND consortium that Frits already mentioned before. And then for looking at bladder cancer recurrence, I used data from the BCPP cohort. So quickly, this is very simplified, I have to say, but what is then the whole hypothesis behind that diet could potentially influence the risk or recurrence of bladder cancer? So the whole idea with fruits and vegetables, for example, is that the more fruits and vegetables you eat, the more antioxidants you get into your body, the less inflammation you will get, and therefore less cell damage, which will hopefully then lower your risk of bladder cancer. But this is actually true also for other types of cancer. But for bladder cancer, there is also this other hypothesis that's very specific for bladder cancer on fluid intake that actually would suggest that if you consume more fluids, if you drink more, that you dilute potential harmful metabolites, like for example from cigarette smoke, that linger in your bladder, that you would dilute them more. And also if you drink more, that you would urinate more and then flush them out faster. So in that way, drinking more would mean like less exposure to the bladder urothelium and therefore also potentially lower your risk of bladder cancer. Currently, the recommendations set up by the World Cancer Research Fund that were done in 2015 and even revised in 2018, actually there's not that much known at this point when it comes to diet. So there is like, yeah, this probable strong evidence at the moment that arsenic in drinking water will increase the risk of bladder cancer. And there is also some limited evidence that vegetables and fruit and potentially tea could decrease the risk of bladder cancer, but it's still very limited and not really conclusive. So for that reason, it's good that studies like BLAND are here to really pool large studies to see if these results can be confirmed and even hopefully strengthened. So for BLAND, BLAND contains a lot of observational studies, cohort studies and case control studies. So for the cohort studies, you actually include people who do not have bladder cancer, and then you look at like, do they eat a lot of fruits and vegetables? Do they eat like more fruits and vegetables? And then over time, they are followed up over time, and then they have a look like who does or does not develop bladder cancer. For the case control studies, it's slightly different. So then you have like people with bladder cancer and people without bladder cancer, so cases and controls, and then you ask them back, like, what was your diet for, say, like a year ago? And then you do your analyses. So the conclusion of what I did, like within the BLAND consortium, I looked at fruit and vegetable intake, and the conclusions that we could make from the cohort studies that if you eat more vegetables, that you probably have a lower risk of developing bladder cancer, especially for women in our study. And for the case control studies, we also found that again, eating more fruits and vegetables, in this case now fruit I'm first talking about, would potentially lower your risk of developing bladder cancer. As I said, I was already saying fruit and vegetables, but it is true actually also for vegetable intake. So-called inverse associations could be found, as in the more fruits and vegetables you eat, potentially your risk of bladder cancer goes down. But okay, so I looked at total fruit and vegetable intake, which is very important because fruits and vegetables are those food items that you expect that have like very beneficial compounds that could lower your risk of cancer. But it's also very important to look at like what do you eat like overall. So look at your whole diet, not only one specific food group. So other students in the group of where I worked at, they also looked at diet. So for example, the Mediterranean diet, and they could also see that if you adhere more to a Mediterranean diet, so a diet full of fruits and vegetables also, that you also could potentially lower your risk of getting bladder cancer. While eating, like adhering to a more Western diet, so eating like less fruits and vegetables and more like junk food, that could potentially increase your risk of developing bladder cancer. Other recent studies, so besides the analysis that I did in the BLAND consortium, other studies also found that with the Mediterranean diet, there would potentially be like a lower risk. Adhering to the Mediterranean diet, you have like a lower risk of developing bladder cancer, or actually that there was that they couldn't find an association with it. And the same goes for like studies looking at the dietary inflammatory index, which is like comparable like with a Western diet. They also found like no associations, or that it would indeed increase your risk of bladder cancer. And a meta-analysis on total fluid intake, so trying to confirm that hypothesis of drinking more, lowering your risk of bladder cancer. Actually, they found no association, so there's no evidence proving that. Well, now going on to bladder cancer recurrence, when I just started my PhD, I actually thought like, oh, that would be interesting to write a systematic literature review on what is known now in the literature from diet and bladder cancer prognosis. And actually, I came to the conclusion that there was only like 4 studies for studies looking within bladder cancer patients at dietary intake. So it was very, very limited. Eventually, I even wrote my systematic literature review on multiple cancer types just to have like a full article. But that was like the first time that I really realized like how important cohorts like BCPP are. There was actually also a group in Nijmegen, also from the Netherlands. They also realized that looking at only diet, like within bladder cancer patients, it's very limited. So they actually also included like body mass index and diet-related factors. And they also came to the same conclusion that evidence is very limited. That there was only like a very small role potentially for body mass index in prostate cancer recurrence. So these are actually the first studies back then that I found that did have a look at dietary intake and the outcome of bladder cancer. So there was like one study on fruits and vegetables intake that found no association with bladder cancer-specific survival. One study on dietary intake of vitamin A that found no association with bladder cancer recurrence. One study, it's a very famous one, that looked at total fluid intake, but also they found no association with bladder cancer recurrence. And one study on alcoholic drinks, showing there was an inverse association with all-cause mortality. So all with all, it was it's really important that more studies are conducted and investigate this within bladder cancer patients. So this is why I took the data from BCPP. In the BCPP cohort, so patients with bladder cancer were included, but at that point, they did not have a recurrence yet. It was like right after diagnosis, they were asked to join the study. And then they were asked like what their diet was like the last year. So we had like data on diet at that time point, at baseline, but also 1, 2, 3, 4, and even 5 years after diagnosis, regardless of the fact if they developed a recurrence or not. Well, and the results, the conclusions from that was, I looked at total fluid intake, and there was no association with non-muscle invasive bladder cancer recurrence. And also when I had a look at total fruit and vegetable intake, there was also no association with non-muscle invasive bladder cancer recurrence. Other more recent studies, they actually also found that if you, well, they didn't look specifically at fruit and vegetable intake, but more like at the whole pattern of dietary pattern, high in fruits and vegetables. And they actually also came to the same conclusion, that there was no link, no association between eating more fruits and vegetables and a recurrence of non-muscle invasive bladder cancer or prognosis. But they did actually find that a Western diet, a high adherence to a Western diet, could potentially actually increase the risk of non-muscle invasive bladder cancer recurrence. However, they did not find it with progression. So at this point, it is still limited, the evidence, but we are getting there. And at a certain point, a lot of people even say like, oh, but if it's like with bladder cancer, smoking is such a strong risk factor, why do you even look at diet? But well, then I think, you know, once you get like the diagnosis of cancer, which is horrible, I mean, you lose all control. And then I think like even if it would be like only 1% that diet could potentially contribute to lowering your risk of like a recurrence or progression, I think that would be already very valuable. Plus it is something that somebody has control of themselves. So I think like also well-designed other prospective studies are really needed to develop more evidence-based guidelines for survivors. Yeah, that was it.
Thank you. We have time for one question, really, because we're behind. Sorry. Shall we take the one that is online?
Well, actually, it was from me.
Oh, so
There's not one online.
Because you've been nice and reading everybody else's question. You get to ask a question.
I was just wondering, in the like ones where it's looking at the risk of bladder cancer, and it was the Mediterranean diet had an inverse effect association. What was that being compared to? Was it just any other diet other than the Mediterranean diet, or?
No, it was like within the people adhering like very well to the Mediterranean diet or like not.
Okay.
So, yeah.
Okay, we can have one more question. Yes.
Was there any difference in, or do you know now with the cohort being larger and tracking for longer, if there's differences in things like mutational burden associated with differences in diet? If it's just a general additional, do people with a worse diet have just more mutations in their tumors or anything like that?
That could very well be possible, but to be honest, that's not my field of expertise. I couldn't tell at this point. No.
Thank you.
Thank you. So then our next speaker is another real-world evidence expert. So this is Nadia Lipanova, and she's going to tell us more about germline prognostic markers for bladder cancer. Thank you.
Hi, everyone. Thank you very much for having me. And it is an absolute pleasure to be presenting my work on germline prognostic markers and urinary bladder cancers. And no, please don't go. There we go. And it was entirely carried out within the BCBB. So I'm preaching to the choir here, but most of you are really aware that a lot of bladder cancer cases present as non-muscle invasive bladder cancer cases. And at first look, it's good news, because it has relatively good survival, but of course, it takes a lot in the recurrence rate, with almost 70% of patients having at least a single recurrence in the first 5 years. And that in itself is an immense burden on healthcare systems, because monitoring is incredibly costly, it's laborious, and it's also difficult to maintain through times such as COVID. And it's not easy just to do it already. And the main burden is, of course, absorbed by the patients, because this is terrifying to live, let's say, for 5 years and think, oh, I'm probably going to get a recurrence of cancer. So mental health is also a very big problem among these patients, even if survival is not that big of a concern. So at this point, both patient and the clinician have a bit of a decision to make. On one hand, say if you knew that you had a recurrence of your cancer in the next 2 years, or if it would progress, or if you would die down the line 3 years, you might as well be aggressive right from the start. Through all the means that you have, maybe even cut out the bladder, because you want to increase your chances of the better outcome. Now, on the other hand, what if you knew that you will never have a recurrence, or maybe cancer will never progress? So you don't want to also subject the patient to overtreatment, and that's a lot of burden in itself. This is really keeping me on my toes. The problem with this decision-making is that there are very few tools to help decide. And there are some, they're quite good on population level, but once we consider a single individual patient, it's losing its precision quite entirely. So the sole question of my work and my PhD was whether genetics can help predict the outcome and aggressiveness of the bladder cancer, to pretty much help and make that decision a little bit easier. Before we wanted to contribute our own work to the field, we of course had to take a look at what's already been published, and there was a lot, and there was a lot of buzz. Notice that there was also a B motive in Lydia's presentation, which is a coincidence, but a lucky one. Specifically, how much was out there? There were 112 studies, combined reporting 316 associations, which is far more than I've expected. And if we look at the broad picture, we can see that those 360 associations, each representing a single point in genome that is passed on throughout generations, they fall into either prognosis, comprising outcomes of progression, death, and recurrence is, of course, by far the most popular one. And the other domain focuses on disease at the time of diagnosis, focusing on stage, grade, risk groups that are clinically used, age, and tumor size. So they use it as a proxy for what is going to happen and what is the risk of adverse outcome. What do those 316 associations say? First of all, a little bit encouraging news is there are emergent patterns, mainly 3 genes, show to be associated with multiple outcomes. So OGG1 showed to be associated with age, grade, recurrence, risk group, and stage, so quite a few. And at the same time, a duo of TP53 and MDM2, so they travel together, were associated with death, grade, recurrence, risk group, and stage. And the interesting point here is that both sets of genes are very pleiotropic. They're not specific to bladder cancer, probably itself, as of course TP53 is the most famous cancer gene, but they are associated with a lot of processes. So they are involved in biological mechanisms that overlook the overall DNA preservation, and hence they're also not specific to any outcome. They just show that a lot of things have gone wrong. But overall, despite the few patterns that we have seen, the genetic distance between the outcomes is very large. So if we take an example of recurrence and death as to one of the most important outcomes, recurrence has all of those genes that you see on the screen that are completely independent from the genes that were associated with death. And death in itself has a completely different set. So they are quite independent and they are quite different in terms of what biological mechanisms are involved in developing each outcome. So at this stage, first thing that we realized, there was a considerable heterogeneity between study designs, outcomes, statistical approaches, and reporting, which is a horribly boring point to make at this stage, but it showed probably a lack of consensus that we did not expect in the bladder cancer community. And I think it stems from genetic epidemiology becoming very popular in the last, now 15 years, and everybody rushed to discovery stages. Everybody reported a lot of discovery steps and genes, but they were not correlated. It was really difficult to compare studies and draw one conclusion. The second point that we realized, the clinical phenotypes do point to different biological pathways. No matter how much you shift those genes and outcomes around, they do seem to be representing different biological, pathological pathways. And that is really important, so we can't just muck up bladder cancer outcomes into one. And then finally, what we've noticed, there was not a single genome-wide association study at the time performed for prognosis or baseline characteristics. So those 112 studies, each of them had a hypothesis. They went after a specific gene or after a specific region in the genome, and they tested that. But nobody took a step back and said, okay, how about we let all of those bases tell us what is actually significant? Which is exactly what we've done. So we've gone to a bladder cancer prognosis program. We've only focused on non-muscle invasive bladder cancer in this case. And each of those patients, about 650, contributed about 12 million markers. And each marker is a very minuscule point in the genome. And there are 3.2 billion in each one of ours. And out of those millions of markers, we found 61 SNPs that have reached genome-wide significance, which is just penalized significance statistical level. 47 of those were associated with tumor size, and then some were associated with age, stage, and grade at the time of diagnosis. Now, we've then moved on to replication stage, and we've gone out, found a completely independent bladder cancer program cohort, in Nijmegen, which is in the Netherlands. We had a few less patients and also not an entire set of our genetic regions to be tested, which is unfortunate, but that happens a lot. And then out of all those millions, we had only one, the chosen one, that was significant in the meta-analysis, and it was associated with tumor size and is located on chromosome 13. Now, this is a bit of a closer look on chromosome 13, so you can see a nice tail stemming from the SNP that we have found to be significant, which is really good news, because you don't want to have a completely independent SNP and the whole region surrounding that SNP not to show any statistical significance. You want to be confident that it's showing that the whole, hopefully, gene region is also associated. Now, if we're going to zoom in a little bit further, we can see the same thing, and each dot represents that single SNP in the genome, and we can see that there's quite a lot of inflation. One of the most important things that you can see here is in the bottom table that our SNP of interest falls within a gene, and that's NBEA. NBEA was not known in bladder cancer, or was not published well, let's say, but NBEA is very well known in neural development. Dramatic mutations or knockouts of NBEA gene usually result in severe neural well, not disabilities, usually ends up in pretty much death of a fetus. So unfortunately prevents any kind of neurodevelopment. And then milder, some point mutations usually result in learning disabilities and some other neural phenotypes. It is mainly explained by the fact that synapses without full set of NBA protein cannot grow. They cannot properly grow, so they don't connect where they're supposed to be connecting. And at the time, this gene was not known in bladder cancer literature, but since we've published, I think it was quite a few years ago, it has come up again in bladder cancer, so there's some promise of that. Now, at this stage, I've realized we have two big problems, which we're concerned a lot about. And we had very few replication studies. So all the premise that we were building our case on is already questionable. There's a lot of discovery, not a lot of replication. The other problem was that there are few bladder cancer cohorts with clinical and genetic information. So you kind of introduce a catch-22 problem. You don't have any data, you won't have replication. And if there's not replication, people are not going to start those initiatives altogether, which prevented ourselves to validate any of our findings. It was very difficult to do. And at this stage, we realized that we needed to be crafty with our approach, which is exactly why we went to UK Biobank. I'm sure all of you know this resource, but it's quite rich. It has a mixture of data, electronic patient records, genotyping, death and cancer registry data on about 500,000 patients, which is a lot. And at the time, it had about 1,500 bladder cancer patients, which was amazing news for us and for our work. Now, all of you that have worked with real-world data are, of course, questioning, yeah, that's great, but what about non-routinely collected endpoints? You can have diagnostic information, all is fine. Where's the recurrence? Where's the progression? None of these are recorded, hence you can't do that. Well, to this, we can see the patient's trajectory is reflected in recorded events, when based on these, you can actually build reliable proxies that would show you who has progressed, that would show you who had a recurrence, and also what was the stage, let's say, at the time of diagnosis. So there's a lot to be read from these databases, which again is exactly what we've done. So we went to UK Biobank, used diagnostic procedural information, and then really aligned on the timings of recording of each, and built proxies for recurrence, progression, and invasiveness at baseline. We could not accurately estimate who, let's say, was stage 3 or 4, but we could say whether it was invasive or non-invasive based on the treatment. After we had these, we of course wanted to test them. Are they any good? Which is why we have taken all of the significant SNPs right from the start, the ones that we scavenged from the literature, and all of the SNPs for phenotypes that we have found, we ourselves have discovered, merged them into this big testing set that resulted in age, progression, survival, and recurrent SNPs to be tested. And a few of these have actually replicated, which is always good news, because usually it doesn't. In genetic epidemiology, usually it just doesn't. And we had 8 of those across progression, survival, and recurrence. And the bottom line of this analysis was that we were able to suggest attractive gene targets for bladder cancer prognosis study, which was pretty much a set of 5 genes, which you can see on the screen. And from my perspective, this is the part of work that I'm most proud of, because I think it really merges discovery, validation, and practicality that I think is very commonly lost, especially in genetic epidemiology. Because I think all of us are working for the punchline, and so what? And so what should we do next? But with this, we can actually go and take these 5 genes into further stages of replication, where you don't need to do another discovery study. These are already replicated across at least 2, maybe 3 studies, and that's usually better than what you have with genetic biomarkers, germline, genetic biomarkers for bladder cancer. To summarize, first of all, bladder cancer phenotypes are not created equal, and each genuinely deserves attention. I think looking at study to study, this has been somewhere reserved and somewhere kept, but I think the more we focus on the different biological pathways and looking at the genes and how they explain each is is really, I think, a better path for success of finding anything that is useful for patients and clinical management. Second take-home message, and I'm incredibly biased because my pride and joy and passion in life is real-world data and real-world evidence, but secondary medical data can and should complement clinical trials and prospective core studies. I think we are all realistic. Financial resources, human resources are horribly difficult to source. So we're not going to have another BCPP in the next, let's say, 2 years. But we should, I think, be a little bit more creative with all the data that we have already and how we can potentially enrich that data, how we can use that data in a slightly different way. Because I think there's quite a few initiatives in UK alone, Europe-wide, US-wide. There's quite a lot I think to think about. And then finally, definitely not the sexiest part of research, but evidence is generally built in consistency. And discovery in itself is nothing without replication. And I think I've just mentioned the love of practicality that I think is sometimes lost, especially in genetic epidemiology. And I'm really excited to see some of the efforts built upon different studies that I think a few people discussed already today. I think that's one of the most important things. And with that, I'm very happy to answer any questions we have. So thank you.
Thank you. Any questions for Nadia? Are you all getting a little bit tired? So what do you think would be the next, or like if you had all the money in the world, what would you do next with, like, you know, you showed your 5, you know, you have your 3 take-home messages. So what would be your ideal approach going forward?
Oh, this is a very sweet dream. Well, first of all, I would definitely take those 5 genes and take those into molecular replication. One of those, actually, I forgot to say, RGS5 has been already shown to be significant for bladder cancer in another study, which I'm really happy about. So I would probably focus on RGS5 and the rest in molecular, and then hopefully, eventually, go into further studies that would be costly, but I would have all the money. So.
Thank you. So we have one more speaker, and then don't run away, because we still have prizes to award too. So Roland Arnold has the pleasure of giving us the last talk for today about novel genomic phenomena as bladder cancer biomarkers. Thank you.
So, first of all, thank you for giving me this opportunity to speak here at this excellent event. So, yeah, I'm Roland, obviously, and I'm the theme lead for bioinformatics and genomics for the Bladder Cancer Research Center in Birmingham. And don't fear now something too complicated. I really just want to give you some outlook of what we're currently working on. And the title I put into the program was a little bit of a lie, I noticed. So the first lie is, I said genomic, that's too general. I'm more interested into transcriptomics, which is actually the active genes, what happens there. Then I said, second lie, novel. So I'm not necessarily interested in something which is completely novel as a concept, but the things which we perhaps neglected for too long. And then the third lie probably is that I somehow suggested I have a new biomarker, which is not true. We are working in that direction. So I like to say, to make it interesting to say, it's a dark matter of transcriptomics I want to look into. So now I hear this. Right. So you all know this. I have this slide for today just to remind myself or somehow to motivate. So this is I'm a bioinformatician by training, so I have to remind myself about this. But this is my coordinate system, or our coordinate system, where we work in, if you want so. So we have the different stages of the cancer, the different grades, they're differently dangerous, and they definitely have different subtypes. And our aim somehow is, as data people, if you want so, can we basically predict or see something which is special for the different stages or grades? And perhaps we can do so by looking at unusual molecules. And And there's a usual motivation why we want to have biomarkers in the future, why we want to have to stratify patients. So I want to illustrate now the way I try to think. So on the left side, you see basically a plan. It's a subway plan, and this is something like the DNA. So that is what most people look at if they do bioinformatics or genomics. I have my DNA sequence, and I'm looking for mutations, and that's it, right? Of course, that's very important because we know cancer is a disease of the genome. But what actually happens from day to day, there might be a traffic jam, and that's basically, if genes go wrongly expressed, so basically not this whole plan is active. So depending on the cell state, cell type, and so on, different stations are activated, if you want. So that the other genes. So many people now look at the traffic jam or compare the traffic between two states, which is a differential gene expression analysis. But what I want to do is to look really for these unusual guys here, like this very strange car, and see, is it of any use or any interest? Okay, so the first big area we are working in is actually isoforms and splicing. So as probably most of you know, so the gene is encoded in the genome. In eukaryotes, it's organized in exons and introns, and then it's expressed, transcribed into a messenger RNA, and during that process, it is spliced, so the introns go away, but it can be alternatively spliced, generating different isoforms. So what often happens in cancer, what I always say is everything does and can and does go wrong in cancer. So it happens that we have missplicing. So basically, it's a normal splicing program is not working properly for whatever reason. So it might be generally or punctually, and gives me some of these, which I call local events. That we have stuff like exon skipping. So an exon which is normally used, it will not be used. Things we call mutually exclusive exons, so only one alternative appears, and that was not in the normal state, and we can have change of the boundaries or even intron retention. So, so these are basically two things that we somehow try at the moment to exploit. Which defines somehow our research directions. So we're trying to establish existing and novel tools to mine these splice variants as biomarkers. So there are two ways to do this. So one is an isoform stronger expressed as normal, and the other way are these small novel events I showed, like alternative donor and acceptor sites. So we have sometimes very difficult and complex isoform families where it becomes, with the sequencing data, very difficult to distinguish what's exactly going on. And we have a very nice PhD project on this. So we started now going, also not only looking into, the not so novel stuff like isoforms, but using new technologies, especially long read sequencing, because only the long read sequencing could give me the actual real isoform. The short read sequencing, I need then the bioinformatics approach to reconstruct how the isoform might look like, but I'm not really sure. So that's going on, and we have several student and PhD projects with it. And then we further on also want to see not only biomarkers, but can we also say something about cancer biology? So, for example, these splice forms might give neoantigens, which might explain some interplay with the immune system. And then I have in bold here this the thing I want to present to you now is something which is really strange. What sometimes happens is back splicing. So splicing has a direction, and sometimes what simply happens is that it changes direction and splices back and forms a circle, and forming something which is called a circular RNA. And so that circular RNA exists, is known for a long time, but people quite ignored it until recently. Now it's totally getting off as a topic. Not so much in the UK yet. I think we are still spearheading, looking into it. But what people, so first people thought this is just transcriptional noise and doesn't mean anything. But now for individual instances of circular RNAs, it's now shown that there is a molecular function. So some, and often molecular functions which are specific to cancer. So they might interact with basically with regulation. For example, some of them sponge ribosome RNA binding proteins or microRNAs and takes them out of the system if you want, so they couldn't interact with the linear form of the messenger RNA, and so somehow they do influence things. So just a short summary now. What is interesting on them? So they're single-stranded, really like circles. They're very stable because messenger RNA degradation happens at the ends, and obviously the circle doesn't have an end. So that's interesting because the RNA per se is not stable and degrades quickly, and the circles might be, you know, something which stays longer and might be found then or is found also in body fluids like blood and urine, especially. It might therefore be a theoretical biomarker. And it somehow all happens during splicing. So the circles have their own inner structure of introns and introns spliced out, mostly, not completely, which is all interesting things to look at. Okay, so does it matter? So do circles exist only in 1 or 2 copies? Now there are many. So this is from a review. It's already an older review on the left, which just shows you the numbers of how many different individual circular RNAs, not copies, really different coordinates, have been found in different cell lines. So it's many. On the right, without going into much detail, because then I speak for too long, this is our first study on circular RNA, where we basically just in the BCPP cohort, where we had 57 deeply sequenced samples where we could fish for circles. So it's RNA sequence samples. So basically, depending on which software to detect them, we found basically more than 3,000 individual different circular RNAs in these non-muscle invasive bladder cancers, which behave like you expect. So most are spliced, but they're circles. So in short, this is what on the right side. So this is, we made this nice small publication. So this is my most complicated slide, don't worry. And of course, because I'm a bioinformatician, I have to show a heat map. So what it shows, what it wants to show is, so we, in a supervised fashion, looked for circular RNAs which are independently from the linear form of the messenger RNA, differentially expressed between different grades of the tumor. And this one is just, if you want the heat map, is just an illustration how well that worked. So on the x-axis is, of course, too small for you to read, are basically the coordinates and the gene names where the circles come from. And on the x-axis, the color code, these are the grade information of the tumors. And then basically clustered this to see if there are tendencies. So, but before I explain, we can now, I don't know if I have a laser pointer here. It should be, right? Yeah. It's very faint. I don't know if you can see it. So there's this interesting group here of G3, which clusters separately from the rest. And this is only, and then in the circles, this comprises something, a low expressed group of circles, which is high expressed here and inverted here if you want. So basically there's 3 clusters of circular RNAs. So on the right here, this is just, I put the genes where the circles come from into a database which spits me out how these genes interact. And so this big blob here, this is all cell cycle related genes, but that is basically turned off in terms of circles here. So the assumption now is, which you could test in the future, that these circles here influence somehow cell cycle, and this only plays a role in that set of cancers. So then this bit here is very empty with interactions. So basically these circles act in completely different molecular functions, and it's completely unclear, but they act in a concerted manner or regularly in our samples. And here's just some cell signaling, but also not much. So that is basically like, like a snapshot. How does the circle look like in non-muscle invasive BCPP samples? Yeah, if you want to read more of it, it's a small article in Frontiers of Oncology. So there's a little bit more now, an outlook if you want so. So what we're always lacking is real normal tissue to investigate all this. So the problem is that if people do transcriptomics, they only send in the tumor for sequencing, but no matched normal. So this is really a problem, but we found a few paracancerous tissues from a Chinese group, which have been processed in the right way in the sequencing, so we could look for circles. And you can see that this was paired with cancer samples, obviously. And two things to say here. So one is that there are circles which look private to the cancer. There's a large overlap. Difficulty is this really normal? We don't know. And there's even something which perhaps is like a circles which only show up in normals, but are then lost in the cancer. But this is a very small number of samples. I think it's in total 10 samples. So I urgently try to find more, paired tumor normal pairs and money to investigate this further. Also, some of the circles in the cancer are much higher expressed, quite highly expressed compared to the linear form. So only thing I want to say here is there's massive room of further investigation, which I aim to do, or we aim to do. So this is already the end. So I don't know, I just want to mention everybody who is involved. So we work very closely with our proteomics and biomarker group. So there's Doug, who is there, and Nahima, who's actually doing a lot of, always if I want to have sequenced something, she has to do work. So thanks a lot that you always do this. So then Anshita, who's unfortunately not feeling well today, I think she might join us online. She did all the circular RNA work. And then we have Minghao and Nada, who are working on isoform-related projects. So Nada on this mentioned thing of detangling a very complicated family of isoforms and to check if they're potentially good biomarkers. So in that project, we also try to figure out what are the best ways to work with these isoforms. So because it's if you say an isoform is a biomarker, it's very difficult to measure. But what you can, for example, try to measure is the relative expression of two isoforms to each other, and that might be unique for a certain cell state or cancer state. And Minghao started now working on checking out if the novel isoforms can tell us something about interplay with the immune system. So that's everything. And yeah, and with this I end. And if you have questions, please ask.
Thank you. No. We'll get you questions. We're not going to let you walk off. No. Any questions for Roland? Oh, come on, we have to ask him a question. Yes. Oh, there's even two. Excellent.
Hi, Roland. So what are they doing? You've got these collections of ones that presumably they can't be translated.
Yeah, so that's a good question. So originally I thought none of them can be translated. A few, but I've then found in the literature information that a few of them can be translated with some strange viral entry translation initiation site. But this seems to be only a very, very small minority of them. Would be interesting to see if we could do something with mass spec on it, but I didn't think this completely through yet. But we could discuss later more about this.
Ananya has a question too.
Thank you. Really interesting talk. So there's lots. I mean, lots of us are doing work on gene signatures. You've also got gene signatures in the urine. My group's done quite a lot of work on transcriptomic signatures, on FFPE. How do you bring it all together? Is it microRNAs? Is it normal RNA? Is it circular RNA? What should we be focusing on?
I really don't know. So I think this, standard bioinformatician answer would be integrate everything into one model, but I think that's not really working. And I think probably different molecule types are differently good for different questions. So from FFPE, for example, it will be very difficult to do real, isoform work because it's so degraded, the RNA. So there you likely want to go in with, this mutation. If you look at urine, then that's a big question. So I hope that the circular RNA shows sufficiently up in urine that might be useful, but I cannot say for sure yet.
So do these cRNAs exist in any other guise? Do those actual sequences, are they found in genes, for example, or are they regulatory and elsewhere? So is the fact that they're circularized part of just something that happens?
Yeah, so this is what people have seen in some instances of them, is that actually the circularization and the fact that they just sit there without being degraded is part of the function. So they attract, for example, then something what's binding to them, and binds to them and is basically taken away, like a microRNA, for example. So some have a lot of, like, by chance probably, a stretch of the gene which has a lot of microRNA binding sites, and this is preferably made into a circle, and then the microRNA couldn't interfere with the normal linear form anymore. But there's so many different one of them that I don't believe that all of them have such a function. It's only a minority. And how I like to look at them is that if their expression is elevated, then basically the cell expresses them rather than the linear form. So it's a little bit like might diminish the linear form. It might be an alternative layer of regulation, but it's just my thinking.
Thank you.
Great. Thank you very much. Now, before I'll give Rick the word too, we have to do the posters and the oral presentations. But before I do that, I have made myself a list of all the people that need to be thanked. And I'm sure Rick might think of other people that I have then forgotten. But before I get there, it'd be really great if you could fill out your evaluation forms, which sit in your pack. And if you haven't done so, please try and fill out our research prioritization survey. Saren sent you another email today. It will just really help us when we write our MRC application to try and get more funding for bladder cancer research infrastructure in the UK. So we'd be very grateful if you could make the efforts to go online later on, maybe, or on the train, and fill out that research prioritization survey. Now, there is a whole list of people that we need to thank for having this meeting today. First of all, I would like a really big applause for Saren, who is in the back there, who has worked with the Birmingham team to really organize this entire meeting. I think it went very well today. So a big thank you to Saren. But also a big thank you to the Hopin team from KCL, because we were all very stressed about it. And I think they've shown us that it is all doable. And I think it all went very smoothly today. And if we do it next time, then we'll make sure that our online attendees also will be on video, and we can work on that as well so that they can join us and speak in the conversation. So big thank you to all of you for today. And then also a big thank you to Frontiers in Oncology, who has given us the prizes for the poster and oral presentations, but also to Merck, because they have given us an educational grant to help contribute towards the cost of the conference, because sadly, just you guys paying for being here will not pay for all the costs of organizing a conference. So a big thank you to Merck as well. And then obviously also a big thank you to all our speakers and chairs, including Nick James, who has been chairing whilst being online, not chairing, sorry, been marking, if you like, the oral presentations as well, and I'll give you the results in a second. And then finally, I would also like to thank all of you for coming here today, because I think we're very happy to finally be in person again and have managed to kind of do this online. And we very much hope to see you also in 2023 in Birmingham. And so perhaps Rick can say something first, and I'll keep you really waiting till the end, and then I'll tell you who won the prizes.
Yes, thank you. And one final thank you from two final thank yous. Firstly to Linda Briscoe at the University of Birmingham, who's very much been Saren's opposite number in liaising between both our teams, and Bridget Bell, who's here, who has supported Linda with that, and obviously is here to attend the meeting. So thank you very much to those two individuals. No specific plans really to reveal for next year for Birmingham. We will ideally have the meeting in Birmingham and maybe the same venue as we did back in 2020 at the university's hotel, the Edgbaston Park Hotel. But no details yet. But I think it'll be a similar time of year, early March, and yeah, hopefully as many, if not more, attendees. And again, just to reiterate, Mika, thank you to all our speakers and chairs. It's been a great meeting. Thank you.
I'll say who this and you can give it. So, the second prize for best poster goes to Doug Wart. You can come and get it. It's very official. And the first prize for best poster presentation goes to Benjamin Murray. Oh, you'll go there for Saren's poster.
Oh, yes. Oh, yeah.
And the second prize for best oral presentation goes to Simon Baker, who is in the virtual world, but we'll just give him a round of applause. And then finally, the first prize for best oral presentation goes to Chi Kin Ten. Congratulations. So thank you all for attending, and we look forward to seeing you next year. And don't forget the survey. And that you can leave it.